All right, I started handing back the content checks. I will continue to hand them back if you haven't gotten one from me yet, when we do some of these group reviews. One thing I noticed from, I think this section of my lecture, I have 3 people that turned in bonus points with no name. So if you recognize your sheet up front and would like to claim your bonus points, let me know. I have a list of those that did. I'm going to do my best to get you those points, but there is no name, so I couldn't do anything with them. All right, I had a question come through, and I've answered it on the slides here. So, no, I don't expect you to know concepts that we haven't gone through. But if I ask you to watch a video and then we talk about those concepts, make sure you know them, right? Because we did talk about them in class. Let's see. All right, so today we'll keep talking about organelles, and we'll move into cell membrane transport. I'm actually going to assign you a video tonight to watch that will end the cell membrane transport. And then that'll be the last of the content you will be tested on for exam 1. So if you look at our schedule, you might see that on Tuesday I had scheduled a lecture about enzymes and energy. We might talk or start that lecture about enzymes or energy, but I'm not going to test you on it for exam 1. That will be tested on for exam 2. Right? I figured that it's a lot of content, and I feel like I've just not given you enough time between Tuesday and Thursday to absorb any of that new material. So if you downloaded the study guide a couple days ago, you probably have questions pertaining to energy and enzymes. I've gone back and edited those and put a note saying I will not test you on them. Just be mindful as you're studying. All right. Let's jump in where we left off then. All right, on Tuesday we talked about organelles, right? And we talked about the function of organelles, but we did not talk about all of the organelles. There are 2 more that we didn't talk about because they fall in this unique category, in that we call them semi-autonomous organelles, right? Semi-autonomous, meaning that they can grow and reproduce themselves. They also contain their own DNA, right, which is very unique for an organelle to have. Right, now, if they were completely autonomous, they would be able to exist by themselves on their own, would not need help from anybody else. But we call them semi-autonomous, right, because they do rely on the cell for certain molecules or certain parts, right? So the cell will provide these organelles with some of the building blocks that it needs in order to grow and reproduce, right? So it does rely on the cell. But they are unique in that they grow and reproduce, right, on their own, and they have their own DNA. So we're going to talk about 2 of them. The 2 are mitochondria and chloroplasts. And we're just briefly going to go over what they do and their functions, because the next unit we will explore the entire time that we have for that unit on mitochondria and chloroplasts and their functions. All right, so they're semi-autonomous. They carry out similar types of functions, slightly different but similar, in that they are energy converting. They will take in a resource and convert it into an energy source. Right? Mitochondria, they carry out cellular respiration. Right? So they're taking in energy in the form of, most of the time, sugars, breaking down that sugars and giving our cells ATP, right, which is an energy currency of the cell. And so ATP basically carries energy within its molecule. Like I said, we will elaborate on this next unit. we find mitochondria in both plants and animal cells. Right, so both plants and animals have mitochondria that convert sugars into ATP. Now, only plant cells, or mainly plant cells, there are some exceptions, right, will have chloroplasts. Chloroplasts have a similar function. They're taking in a resource, they're making energy out of it. But now chloroplasts, they are taking in energy from a light source, so sunlight, and then converting it to sugars. Sugars which then can then be used to be broken down to make ATP. Right, so chloroplasts, we really only find them in plant cells. Mitochondria, we find them in plants and animals. And next unit, we'll talk about how in plants, the chloroplasts and the mitochondria work together, right? All right. I'm trying not to go too fast today. I'm all hyped up because I have like 20 slides I need to get through in order for you to continue your content checks. But my last lecture period, we did that with no problem. All right, I mentioned that mitochondria and chloroplasts have their own DNA, but I didn't mention why. Right? I want you to think about this, Google it with your neighbor, talk about it for a couple of minutes, and then we'll come back together. I think you might have submitted yours online. Yep. I want to check in with you because if I didn't, we would. Thank you. I don't know why I'm walking behind you all. I want to see your faces. I struggle with this class. I don't know why it's this class or this lecture section and not oh my, Jordan. Jordan, okay. Here. I will type my own things. Thank you. It's on the file.
Really good on
All right, what did we find out? Why do these organelles have their own DNA?
And then they can produce their own proteins, some of the things that they need right there.
Mhm.
Instead of having to ask the nucleus.
Yeah.
Information's there.
Yeah, so their own DNA allows them to kind of work a little bit more independently. Why does it have its DNA, like how did it get its DNA in the first place?
It came from prokaryotic cells.
Oh, came from prokaryotic cells. Did we have something similar?
Well, yeah, they serve as bacteria.
Yeah. So these cells, they have their own DNA, because if we flip to the next slide, because of something called the endosymbiosis theory, right? The theory is that mitochondria and chloroplasts, they were prokaryotes to begin with, right? They were bacteria cells living on their own, right? And then at some point in their history, these cells, these eukaryotic cells, engulfed those bacteria cells. And rather than these bacteria cells being digested or broken down, they actually started living within the eukaryotic cells, right? And when they started living with the eukaryotic cells, both the prokaryotic and the eukaryotic, found out that it's actually a good relationship, right? They exhibited endosymbiosis, right? So both parties are living with each other, coexisting with each other. Specifically, there's a smaller species living in the larger species, right? The cell had the benefit of reaping the rewards of, say, chloroplasts and mitochondria, because they are energy converting, right? So the cells got a lot of energy out of the deal, right? The prokaryotes, mitochondria, the chloroplasts, they are now living in an environment that provides them directly with the resources that they need to live, to grow and reproduce. They don't have to go search out those resources, right? So it worked well for both of them, and then eventually, right, it just became the eukaryotic cells that we know today.
So does that make it a mutualistic relationship?
Yeah, we think it started out as a mutualistic relationship. Yeah. So that's the idea of why they have their own DNA, because we think they used to be their own individual cell, and then started living with these eukaryotic cells, and now they coexist together, right? We don't get one without the other. All right. So we have talked about different parts of the cell, right? We've talked about the nucleus, right? Contains the DNA, controls what proteins are being produced by essentially allowing parts of the DNA as RNA to leave the cell and give instructions to the ribosomes. We talked about the endomembrane system, right? So all the organelles that kind of work together within the cell. We just talked about our semi-autonomous organelles, so our mitochondria and our chloroplasts, right? Ones that are slightly more independent, right, but still rely on the cell. And we also talked about the cytosol, specifically in context of the cytoskeleton, right? Those motor proteins, those protein filaments. So we have all parts of the cell working together to make it alive, right? Give it its function. All right. So if all parts of a cell, or if we tend to have all these parts in a cell, why do we get cells that look so different from each other, right? This here is like a cheek cell versus a neuron cell in the other image. They look vastly different, right? So how do we get these differences? Particularly because they contain their own set of DNA, or because they contain the entire set of DNA in every single cell.
Well, it's because only certain parts of the DNA get replicated or transcribed into proteins, and the proteins make the cell unique to what it is. Because all cells, they share the same DNA, so any cell could become any cell. It's just that it has a specialized function because of what DNA is getting used to make proteins.
Yeah, that's exactly it, right? So all cells have the same DNA, the exact same DNA, but only parts of that DNA are being read, and only certain proteins are being made, depending on the function of the cell, right? So if we think about all the proteins in our bodies, right, there's something like 400,000 different proteins in the human body. One cell alone has the ability to make all these proteins, but one cell alone is never going to make all the proteins. They're only going to make a subset of these, right? And that subset depends on what the cell needs to function as, right? So we would say they have different proteomes. proteomes are the subset of proteins that are actually made, right? So even though every cell has the ability to make 400,000, right, they're only going to make a subset. They're only going to make their own individual proteomes, right? So this can vary in a couple of different ways, right? So first of all, we're only making a certain subset of proteins, right? So which proteins are expressed, which proteins are made, right? Cells can also vary in the levels of expression, right? So if they're making a protein, depending on their function, they might be making more of that specific protein than a cell with a different function, right? Which subtypes of proteins are expressed, or what type of modifications those proteins might undergo. So after the proteins are made, how might they be modified, or what particular type of that protein is made, right? Again, depends on the function of the cell, depends on what the cell needs, right? So that's how we get so many different cells.
Is a chromosome the same as an exon, where that's the DNA that's leaving the nucleus versus introns that always stick to the nucleus and never get transcribed?
I would say, yeah, it's similar. Yeah, because exons, when we talk about DNA replication, they can leave the nucleus, and that's what the cell is needing to make a protein. So theoretically, every different type of cell type, right, is going to have different exons that get exported out of the nucleus. Yeah, and introns are never read, right? So introns won't be the same in all molecules, so they're just the ones that are never going to leave for that particular protein. Yeah, yeah. I know, that probably doesn't make a lot of sense. It will in Unit 3, I promise. All right. So, this applies when we look at different cells. Right? If we were to look at a cell, it has a very specific function and makes a very specific set of proteins, right? It also makes a very specific, or expresses more of a certain organelle than others based on its function. So I've given you a list here that I want you to work through, and I'm actually giving you the first answer. So I've harvested cells from the organs of a frog, from different organs of the frog, right? And when I view them under a microscope, and I'm looking at their organelles, so a very high-powered microscope, right? I notice that depending on what organ I harvested them from, they have different expressions of different organelles. They might have more of one organelle than another. So if I look at my intestinal cells, right, the function of the intestinal cell is absorption and digestion, right? So we're breaking down a lot of molecules. I would expect to see more lysosomes produced in these cells than maybe the pancreatic cell, right? So I want you to work through the next 3 cell types on this list and tell me which organelle would you see likely expressed the most. Pancreatic cell could have 2, possibly 3 organelles that you could list here. I'll give you like 5 minutes. I'm going to keep circulating around here.
Morning. Dust in my No, I mean, from you need a lot of mitochondria.
Yeah.
Would you buy it? Yeah. I feel like it. So that's end of hours. You're gonna see a lot. That's what I'm doing.
Wait, I actually feel it and I can't. It's not the no, it's the routine.
Let's just talk about. I did ribosomes and protein. See, we were thinking those two, but maybe the you one. Because it's detoxification. We don't have to go in here. Yeah, that's what I was thinking. I don't think we could do it. That's pretty good.
As the one that she got yesterday was there you go. I think anyone's okay. 9 hours. Dang. I mean, mine was probably the same. And adding time to
Because you didn't want to get that.
I remember this. Yeah, I remember encouraging other people to go do it, but I don't think
We're all confident. I don't know how much sense makes. I love watching like well, I've seen the beginning episode of
All right, let's go over these questions here. If I'm looking at a pancreatic cell, and I know the function of a pancreatic cell is that it makes digestive enzymes. First of all, what type of biomolecule is an enzyme? Is it a lipid? Is it?
it's a protein.
It's a protein, yeah. It is a protein. All right, so essentially we're looking for those organelles that synthesize proteins. What organelles did we come up with?
Rough ER.
Yeah.
Wait,
the
is that
rough
all I can say
ER.
on the test?
What was that?
Like I can say rough ER.
Yeah. Yes, if you said rough ER on the exam, I would know what you mean. Yeah. What other organelle though? And probably the organelle that's probably the most prominent. Ribosomes. Yeah, and they're embedded within the rough ER. So ribosomes, rough ER are probably the two answers that I would accept here, right? Because they're both working in synthesizing proteins.
You have to say both.
Why is it not Golgi? Because I mean, like, there's a lot of protein folding that's happening.
And that's true. You probably would see some more expression of the Golgi apparatus. Yeah, because they do play a role in modifying proteins. Yeah. So that could be true. probably not as expressed as the ribosomes themselves because they are the ones making the proteins. Yeah. I also put on mine that you could have said lysosomes. This is a bit of a reach for me, all right? Lysosomes, in that we know we're synthesizing digestive enzymes, so we know we're breaking down complex molecules. So theoretically, we're synthesizing those enzymes to digest materials. The organelle that digests materials would also be more expressed. All right. So our testes cells, they synthesize hormones, specifically steroids, right? So what type of organelle would I expect here?
Smooth ER.
The smooth ER, yes, they synthesize lipids. Steroids are a type of lipid. All right, and our muscle cells, right, they aid in movement, they have high energy requirements, they need large amounts of ATP. What organelle would we expect here? I heard it. Mitochondria, yeah, because mitochondria makes ATP. Yeah, so mitochondria would likely be highly expressed in these cells. All right, any questions? I told my last class this, I will tell this class this. I love this question because you're putting together a few different concepts. You're thinking about what types of biomolecules are these, right? And thinking about the organelles that make the biomolecules. I have been known to put questions like this on the exam, right? So, I like these questions. All right, we've talked about organelles. We're going to start moving towards the outside perimeter of the cell. We're actually going to jump ahead a little bit, all right? So, all cells outside of the organelles, outside of the cytoplasm and the cytosol, all cells have a plasma membrane, right? Keep that in mind. We'll come back to plasma membrane. Outside of the plasma membrane, they all have an extra meshwork of molecules, right? And this differs depending on the type of cell we're talking about. Cell walls, right? These are found in mostly plants. Right, again, there's always exceptions, right? But again, we're talking outside of the plasma membrane. A cell wall functions in that it's extremely rigid and provides a lot of strength. And if we look at the molecular composition of the cell walls, we can see it looks like a meshwork of different long molecules, right? A lot of times we find a lot of cellulose molecules in this plant cell wall. Right? Extremely rigid. In fact, if you were to say cut down a tree and make that tree into lumber and build a house, right? The wood that you are looking at is mostly cell wall at that point, right? Because at that point those cells are dead, right? A lot of the bark, or excuse me, not the bark, a lot of the wood inside of a living tree are actually composed of dead cells, right? But what remains are the cell walls, right? Again, super sturdy, super rigid. We take advantage of that. Animal cells have something similar, right? So animal cells have what's called the extracellular matrix. I often abbreviate this as ECM, right? Same type of function, provides strength, right? Provides some rigidity. It also has some bonus functions of helping with organization, some cell signaling, right? And again, if we look at the composition of the extracellular matrix, we again see it's like a meshwork, right, of different molecules, all kind of meshed or knitted together. All right, if you're looking at these slides from Tuesday, I actually condensed these slides a little bit. I used to talk more about the cell wall and the extracellular matrix. We're running out of time, so that is material that I condensed. So I only expect you to know what is on this slide that I am presenting, even if you see other slides with expanded material. All right, when I was in college, one of my professors gave me a helpful analogy for cell walls. an extracellular matrix. They described it as being like a sweater, all right? It surrounds the outside of your body. It's made up of lots of threads that are all kind of interwoven or knitted together, right? Now, sweater provides some warmth, right? But cell wall, extracellular matrix would provide the strength, right? So that's the analogy I have for these structures. All right, we kind of skipped ahead of plasma membrane, but now we're going to come to it, right? Talked about the organelles, we talked about the extracellular matrix, right? But what's between the organelles and the extracellular matrix, or the plant cell wall, is the plasma membrane, right? All cells have a plasma membrane. All living cells have a plasma membrane, right? They surround the cytoplasm. They surround the organelles within. And we talked about them a little bit when we talked about our lipids, right? Because plasma membranes are composed of phospholipids. So if you remember phospholipids, they contain 2 fatty acids, right? We can see that in our image down here, 2 fatty acids. They are nonpolar, so they form a hydrophobic tail, right? So they are water-fearing. The other end of the phospholipid, right, is this hydrophilic head. So that contains the glycerol and a phosphate group, right? They are water-loving. This part of the molecule loves to interact with water. And we talked about when we put these all together, they form this bilayer, 2 layers of phospholipids, where the hydrophobic tails are towards the interior, right? We can see that here the hydrophobic tails are represented as yellow. The hydrophilic heads are represented as red in this image here. Right. Now, what's important is that these hydrophilic heads and hydrophobic tails and the way that they arrange themselves make the plasma membrane semi-permeable. Right? So it allows some substances in and some substances to never enter. Right? Oh, I thought I had a different slide next, but it is semi-permeable. Right? And we will talk about transport of molecules across the plasma membrane in a few slides. All right, so we have this bilayer. We have this phospholipid bilayer. It is not static. It moves, right? There is movement within the plasma membrane. So we're looking at our plasma membrane. We're looking at our phospholipid bilayer. You could say that each layer, or each leaflet, right, is asymmetrical, right? They do not look the same. What's on one layer is not identical on the other layer, right? Notice that we have, besides our phospholipids here, other molecules embedded within the plasma membrane. We have proteins. We have some carbohydrates, right? We have proteins that are only on one side or only on the other side. We have proteins that span the entire membrane. Right? There is more than just the phospholipids, and each layer is asymmetrical. They are not identical. All right, so we're starting to talk about what I call, or not what I call, what scientists call the fluid mosaic model. Right? So we talked about kind of this mosaic piece of it. They're not identical. Let's continue on this mosaic piece. I mentioned there were proteins that could be within the plasma membrane. And there's 3 basic types of proteins that we will talk about, and really we're only going to focus on one. And the one we will focus on is this transmembrane protein. Right? Transmembrane proteins are proteins that span both bilayers. Right? We see this transmembrane protein here. Right? There's a piece of it on the interior of the cell. There's a piece of it on the exterior of the cell. And there's part of it that's embedded within the plasma membrane itself. Right? It spans both layers. Right? We are going to focus mostly on this protein because this will come into play when we talk about transport across the bilayer. Alright, there's more than just transmembrane proteins though, so I am going to mention a couple of others just to give you an idea of the types of proteins that you could find. Right, another type of protein is the lipid anchored protein. These are proteins that are only found on one side of the plasma membrane. Right, so we see the lipid anchored protein on the right hand side here. Only found on one side. It does not span the entire bilayer, but it's anchored in place by a lipid, likely a fatty acid. Right, so it's anchored into the plasma membrane directly. And then the last main class of proteins you might find on the plasma membrane are these peripheral membrane proteins, right? These proteins are not directly bound to the plasma membrane themselves, right? They are only found on one side of the plasma membrane. They are not directly bound to the plasma membrane, but rather they are bound to another protein that is embedded in the plasma membrane. So in this example here, we see our peripheral membrane protein is bound with a transmembrane protein, right? It's not directly bound like this lipid anchored protein is into the plasma membrane. It's not spanning the entire membrane, so it's not embedded within the hydrophobic area. Rather, it's loosely associated or bounded with a protein that does have that connection with the plasma membrane. All right, lots of different types of proteins. Let's talk about the fluid part of this fluid mosaic model. All right, so not only is the plasma membrane asymmetrical, but it can also move, right? The plasma membrane is not rigid, it's not static. There are several types of movement that can occur, and this movement is most often associated with the phospholipids rather than the proteins embedded within the bilayer itself, right? Proteins are bigger, they're harder to move. Phospholipids are relatively small, right? And there's different types of movements that can occur. So if I use my hand as representing a phospholipid, right? I'm going to use my fist as the hydrophilic head and my arm as the hydrophobic tail. One type of movement that a phospholipid can do is just a lateral movement, right? Just move kind of side to side within the plasma membrane. And we can see that in our image here, we have our lateral movements, moving side to side. We can also have rotational movement too. So now, maybe it just rotates in place, spins around. Right? And we can see that on our image down below. We have some rotational movement. The phospholipids, they could also do what's called a flip-flop, right? They can go from one side of the membrane to the other side of the membrane, right? Now this likely happens less often, simply because this is harder to do, so it requires energy. And we can see that on this image here. We're going from one side to the other side. We need ATP, which is an energy source. Right? We also need the help of a transmembrane protein. Right? In this case, it's a type of enzyme called flippase. Right? So this is not going to happen as much as the other types of movement, the lateral movement, the rotational movement, just because it's energetically unfavorable. The cell has to expend energy for this to happen, right? But it is possible. All right, so we would say the membranes are semi-fluid, right? It's not static. There is some movement, right? But it's not totally fluid, right? It's not going to move like water, right? All right, so we know these phospholipids can move, but a phospholipid can also influence fluidity depending on some other factors. The first of which that we will talk about is the length of the fatty acids. All right, so our phospholipid has our two fatty acids. They can be different lengths. right? They're not all going to be the same. Some are going to be shorter, some might be longer. And it's these lengths of the fatty acid tails that influence how fluid the membrane can be, or how much movement can actually happen, right? If we think about phospholipids, right, and I'll try to do phospholipids here, if the tails are short, they're likely to not interact with the neighbor that's across from them. But if they're longer tails, maybe they do interact a little bit, right? They kind of catch each other a little bit, right? So if they're shorter, more movement can happen. If they're longer, it's going to be a little bit more rigid. Movement can still happen, but it's less fluid. All right, so shorter tails can make it more fluid. So can the presence of a double bond. When we talked about phospholipids, we talked about lipids in general. We talked about saturated and unsaturated fatty acids. Right? And unsaturated fatty acids had the presence of a double bond. It had a bend in the fatty acid. Right? Now this is why it can be important, whether it's saturated or unsaturated, in terms of membrane fluidity. Right? If there is a bend in the phospholipid here, it makes it harder to interact with its neighbor. Right? And I think this image on the top here does a relatively good job of illustrating this. You can see those that have a bend require more space and essentially pushes the phospholipid next to it a little bit further away. There's more of these gaps within the bilayer. Right? So they're less likely to interact with their neighbor. There's more space, so they become more fluid. All right? So double bonds can make it more fluid. Shorter tails can make it more fluid. There's one more factor in play here. Right? And it doesn't have to deal with the phospholipid molecule itself, but rather the presence of cholesterol. Right? Cholesterol can be embedded within the phospholipid bilayer. Right? So part of that mosaic part of this fluid mosaic model. We have more than just phospholipids, more than just proteins. We can have cholesterol embedded as well. Cholesterol acts as a stabilizer. Right? It helps stabilize the membrane. And whether it makes it more rigid or more fluid depends on the temperature. So if we think about a plasma membrane in a cold environment, right, the plasma membrane tends to be more rigid. Right? Molecules are not moving as much. Right? The plasma membrane is more rigid. It's not necessarily a bad thing, but if it's so rigid that now it's no longer flexible, right? Or if it tries to move and it breaks, then it becomes a problem. Right? We don't want to break our plasma membrane. That is really bad for the cell. All right? The cell will likely die because of that. Right? So what cholesterol does is when we embed more cholesterol in the plasma membrane, it makes it more fluid. Right? So that it's not so rigid that it can break. Right? It loosens it up a little bit. The opposite is true for warmer temperatures. Right? Plasma membranes in a warmer temperature, molecules are moving around more often. It tends to be pretty fluid, but it can be too fluid. Right? There can be too much movement. Right? It's too flexible. It doesn't hold its shape anymore. Right? Again, not good for a cell. If it's not holding its shape, right, then the inside of the cell, those contents can spill out and the cell will die. So cholesterol, when added in this instance, right, makes the membrane more rigid. Right? So it makes it so it's not super fluid. So it has some rigidity to it. So it's not going to lose its shape anymore.
So you add the cholesterol to like if it's cold, it makes it better no matter what the temperature is.
Yeah. So it's going to stabilize the membrane no matter what temperature. Right? If it's cold, it'll make it more fluid. If it's hot, it'll make it less fluid. Right? So it depends on that temperature. But overall, it stabilizes. All right, I talked a lot. I want you to reflect on what we learned. I want you to tell me, why is it called the fluid mosaic model, right? Why is it fluid? Why is it mosaic? I'll give you a couple minutes. You know exactly what the fluid mosaic model is. You know the concept. Good. All right, let's talk about this. Why would we say a plasma membrane is fluid? It can move. There is movement. Yes. So it's fluid because there's movement. Why would we say it's a mosaic? Yeah, it's asymmetrical and it's composed of more than just phospholipids, right? It's a mosaic of things. It's a bunch of different things, right? We have proteins, phospholipids, cholesterol. Yeah. Any questions about the fluid mosaic model? All right, then we are going to turn our attention to how molecules can move across this semi-permeable layer. All right, if you are a cell and you are a cell that is living, you have to maintain an environment inside of the cell that might be different than the outside of the cell, right? They have to maintain a constant environment inside. And they maintain this constant environment by maintaining gradients of molecules and gradients of charges, right? So for example, the transmembrane gradient, or often just referred to as the concentration gradient, right, is the concentration of solutes that is higher on one side of a membrane than it is the other side of a membrane. Right, so if we look at the example up top here, we have our molecules of glucose. We can see the inside of the cell has a different environment than the outside of the cell, and that there are far fewer glucose molecules inside. Right? This cell is maintaining this gradient where there are fewer molecules inside than there are outside. Right? And it could be for a variety of reasons. Right? Maybe this cell is really good at photosynthesizing, at producing sugars, and needs to get the sugars to move elsewhere. So not only do cells have to maintain gradients of molecules, but they also have to be mindful of electrochemical gradients. Right? So an ion electrochemical gradient is looking at the gradient in terms of charges. Right? So if we look at this molecule down below, we see that the inside of the cell has more negative charges, more negatively charged, than the outside of the cell, which tends to be more positively charged, or have more positively charged ions. Right, so we have to maintain this gradient of charges as well. And that doesn't mean we're just looking at one molecule that has a charge, but rather all of the molecules that might have a charge. So again, if we look at this example, we actually have two molecules in here, two ions I should say, that have a positive charge associated with them. We have these triangles, we have these circles that are represented as potassium and sodium, right? They are the ones carrying the positive charge, right? And there's fewer inside than there are outside. And then chlorine is the one represented as having the negative charge. All right, so we're still looking at a concentration gradient, right? There's a different concentration of solutes on one side of the membrane than the other, but we're just adding in this extra layer that now we're also paying attention to the charges associated with those molecules or with those ions. All right, maintaining gradients. The cell maintains this gradient because the phospholipid bilayer is selectively permeable, right? It can allow the passage of some molecules and ions, right, and not allow the passage of other molecules or ions. Basically, it gives the cell the ability to choose what it can allow through. And again, this has to do with the properties of the phospholipid itself. Right? The interior of this bilayer is filled with those fatty acids, is filled with those hydrophobic tails, right? And this hydrophobic barrier then is a huge barrier for the crossing of a lot of different molecules, right? So for example, ions, right, so they have a full charge to them, or polar molecules are going to find it really hard to move across the interior of the bilayer, right? Because ions, polar molecules, they are hydrophilic, they are water loving, they are attracted to water, right? They are not attracted to areas that are hydrophobic, right? So this provides a barrier for their crossing. And again, this allows the cell to regulate what molecules can come in and which can leave, right? Giving the cell some agency, allowing it to maintain these concentration gradients. See a lot of pens still moving, so I'm going to slow down. All right. So if this layer provides a barrier, there has to be some way that molecules can still cross this barrier, right? And there are multiple different ways that molecules could potentially cross the plasma membrane, cross this barrier. We're going to talk about all three of them, though today we're really going to focus on simple diffusion. All right. Let's start with simple diffusion. I'll briefly define all three, and then we'll elaborate on simple diffusion. So simple diffusion, we are moving substances across a concentration gradient. Right? We see the image here, right? There are no proteins embedded that we are currently using for this process. It's just simply molecules diffusing across the phospholipid bilayer. Right? Now it's moving against the concentration gradient. So the concentration gradient that you want to remember is that molecules want to move from areas of high concentration to areas of low, because they want to try to equalize concentrations, right? So we're always going to move from areas of high to low, right? That's what the molecule wants to do. So simple diffusion, we're moving from areas of high on one side of the membrane to low on the other side of the membrane, just by simply passing through. Now, not all molecules can do this, which is why we also have facilitated diffusion. Again, we're moving with the concentration gradient, moving from high to low, but this time these molecules need some extra help. They cannot just simply diffuse across, they have to use a transmembrane protein to allow passage. Right, so these would be molecules like ions or polar molecules, the ones that are highly repelled by that hydrophobic interior of our plasma membrane. Right, they will use a channel or another type of transmembrane protein to move across. All right, so simple diffusion, facilitated diffusion, we're moving with the concentration gradient. It's a type of passive transport, meaning that we do not need to use energy in order for this type of transport to occur. Right? The cell does not have to expend any energy. All right, now there are cases though where the cell might need to move a molecule against its gradient. Right? Now molecules don't like to go against their gradient. Right? This is not going to happen naturally. And this is where we get active transport. Right? Active transport, we're moving against a gradient. We can see in this image we're going from areas of low to areas of high. Right? Not something the molecules are going to do naturally. So we have to input an amount of energy in order for this to happen. So active transport needs energy to accomplish this type of transport. You can see energy is in the form of ATP, or energy currency here. Right? Once that energy is provided, in conjunction with a transmembrane protein, now the solutes can move against their gradient, from low to high. All right, so passive transport, no energy. Active transport needs energy. Passive transport can be simple diffusion or facilitated diffusion, so needing a protein. Active transport is always going to need a protein, and it's always going to need that energy. All right, these are likely terms you have heard before, even if it's been a while. Let's just make sure we're all on the same page. So simple diffusion, let's just walk through a couple examples here. So it's a type of passive transport, doesn't need energy. If we look at the top image here, we're following some molecules of this green dye. Notice they're going from areas of a high concentration of this dye to an area of the low concentration. And they're going to continue to do so until they reach equilibrium, until there's roughly equal amounts of that molecule on either side of the membrane. Right? Now when equilibrium is reached, that doesn't mean movement stops. Right? Movement can still happen. We can still see arrows indicating movement here. Right? But the movement is still happening in opposite directions equally. Right? So if one molecule is diffusing across to the right, another molecule will diffuse across to the left to maintain that equilibrium. So there's still movement, right? But equilibrium is still maintained. If we add in another molecule, right now we have some red dye or purple dye here, right? Each molecule is still going to move according to its gradient, right? Irregardless of the gradient of a different molecule. So green is going to move according to its concentration gradient, from high to low. Purple is going to move from its concentration gradient, from high to low, even if that means opposite directions from other molecules. And ultimately, equilibrium will be maintained, will be reached, and we'll have equal amounts on either side. Alright, so simple diffusion, we're just moving across the membrane. Not all molecules can do this, and not all molecules can do this at the same rate. Oh, I forgot my analogy. So, if we were to think about simple diffusion in terms of an analogy, it would be like a fence, right? A fence would be the plasma membrane. Fences normally have some gaps in it, right? So smaller organisms, maybe like this housefly, could easily fly through those holes, those gaps within the fence, right? But larger organisms or larger molecules are going to have a hard time. All right, so simple diffusion. Not all molecules can do this, and they don't do it all at the same rate, right? So some can diffuse faster than others, and it depends on a variety of factors, right? So how big is the molecule? Does the molecule have polarity? Does the molecule have a charge, right? And what is the concentration of this molecule on either side of the membrane? This image is going to be really helpful on your Moodle content check. If we look at the top here, we have gases, right, that are relatively small, just two atoms combined together. They're also non-polar. They're going to diffuse across this membrane fairly quickly. They're going to have high rates of diffusion. If we move down on this image, we start to see molecules that are a little bit bigger, or molecules that are small but they have some polarity. So water being one of these, right? We know water is a polar molecule, but we also know water is relatively small, right? So they can also go through the plasma membrane using simple diffusion, just not as fast as other molecules, like these gases that are nonpolar and very small. Right, as we move up in complexity, or add in some more charges, right, like sugars, some polar organic molecules, ions, we tend to see that permeability decreases, right? It's very unlikely to go through simple diffusion because it's just harder to do. They are more repelled by that hydrophobic region. All right, this is my last slide before we move on to the content check. So we'll have plenty of time for the content check. I wanted to touch on facilitated diffusion, right? So facilitated diffusion occurs with the presence of a protein, right? So simple diffusion, we saw that some molecules just have a really hard time diffusing across. Ions, large molecules, very polar molecules have a hard time. But the cell is still going to need these types of molecules and ions, so there has to be a way for them to go through the plasma membrane. And that's done so using a protein, right? Using a type of transport protein. And I have a video that just kind of illustrates this as we're talking here, right? So these proteins provide a passageway, provide a safe passageway for those larger molecules, those ions, to move across the barrier, right? Because they can't do so on their own, right? So these are going to be ions, larger molecules. We can see they're only moving according to this protein. Now there are different types of proteins, right? And you'll learn about that in the additional little lecture I have posted online here. But again, if we're going back to that analogy of a fence, right? This is like a fence that has a gate in it. All right, so now larger animals or human beings can move across the fence, but they're aided by having this gate that can open for them. All right. So I'm going to stop there because I wanted to give you plenty of time for the content check. I know last time we were kind of rushing through that, and I didn't feel like that was fair. So I have posted the rest of this lecture online. I want you to follow through with the video. I think it's like 12 slides or something like that. And the concepts I want you to know are listed below here. We will revisit these concepts on Tuesday to make sure you understand it, right, before the exam. But this is work I want you to do before then. Right, the slideshow accompanying this is in the Google Drive folder. Right, the video is posted on that YouTube playlist. All right. So as far as homework for Tuesday goes, Now that we've gone through this lecture, you should be able to finish the first part of content check number 2, the part that is on Moodle. In fact, you can probably finish it in class before you even leave. There should be time for that. And then I want you to watch that video. And then the exam is 1 week from today. So make sure you're studying, understanding these concepts, visiting the tutors, coming to talk to me. All right. Content check. It's another drawing one, right? There is evidence to say that if you draw out a concept, you're more likely to remember a concept. So I'm going to have us draw a phospholipid bilayer, right, and add in some features to this bilayer. Right? Once you have drawn this, you can turn it in up front. I do have a few pieces of blank paper if you want some blank paper to do so. Turn it in up front, turn it in on Moodle, whichever method you prefer. Once it's turned in, you are free to leave. And again, if you want to claim these points, right, I'm going to assume everyone is honest in claiming these points. But let me know, because again, they did not have a name, so I cannot give you bonus points.